All right, so for this lecture, we just talked about organic macromolecules, and now we're going to look at things that use those. Okay, at the smallest level, and we're talking about the cell. All right, now remember, in the first lecture, we said the cell is the basic unit of life. All right, and so when scientists, characterize cells, think about their characteristics. They have developed the cell theory. All right. So basically, what it says is that the cell is a basic unit of life, and all organisms consist of one or more cells. They're either unicellular or multicellular, and that the cell is the structural and functional unit of all organisms. All right. A cell is the smallest unit of life. All right. Individual alive, even as part of a multi-celled organism. All right, so the cells that make up your body are living things, even though they work together to make you. Okay, all living cells arise from division of pre-existing cells. So cells just don't appear; they have to come from previous cells, and we'll look at that process later on in the semester. And then the cells contain hereditary material, DNA. All right, which they then pass on to their offspring when they divide. Now, why are cells so small? Okay, they're small because of the constraints from the surface to volume ratio. As the cell gets larger, the volume, if we're saying, well, cells should just be big. Well, as they would get larger, the volume would outpace the surface that would be able to effectively allow the inflow and outflow of nutrients and waste. And so because of that issue with the surface to volume ratio, that's why cells are small. But all cells have a few things in common. Every cell has a plasma membrane, okay, which is a boundary between the interior of the cell and the exterior environment. All right. And that plasma membrane is selectively permeable. And we'll deal more with plasma membranes in the next lecture. But remember, we said in the previous lecture that a main component of plasma membranes are phospholipids. Okay, remember phospholipids have hydrophilic heads, okay, and hydrophobic tails. And so the hydrophilic heads are oriented to either the outside external environment or the interior of the cell. And then the hydrophobic tails are facing each other. So you have this lipid bilayer. All right. And the plasma membrane is said to be selectively permeable. So only certain things can move across the plasma membranes. Every cell has cytoplasm, which is a fluid that is a mixture of water and other organic molecules. All right, and so when we're going to talk about cellular metabolism or cellular respiration in a couple lectures, that occurs in the cytoplasm. The cytoplasm also houses all the organelles that we'll talk about later. And then all cells, like we said during the cell theory, contains DNA, which is the hereditary material of the cell. Now, it doesn't mean that they always keep that DNA. Some cells lose their DNA as they mature. For instance, your red blood cells. Okay, let's see. Just kind of, again, talked about the three components: plasma membrane, cytoplasm, and all cells start out life with DNA. Now, in eukaryotic cells, all right, that DNA is contained within a nucleus. And as we said, most cells are really small, 1 to 100 micrometers in diameter, right? Smaller than what your unaided eye can really perceive. So how do we look at cells? We use microscopes, right? And we'll look at microscopes in a lab later on in the semester. there's two basic types of microscopes. There are light microscopes that use visible light to illuminate the samples. And then, so they use a series of lenses, or one lens, to focus light into a magnified image. All right, and then you can use stains or light emitting tracers to see the details inside the cells. Another major type of microscope are the electron microscopes. Instead of using light, they use magnetic fields to focus a beam of electrons onto a sample. Now, the drawback to electron microscopes is you have to kill the sample in order to use or see that sample with the electron microscope. Right, so here is an image from a light microscope. These are various species of diatoms. And then here is an image from an electron microscope. Electron microscopes can see more, have a better magnification than light microscopes. All right, so they can see smaller things at a much more magnified level. Now, when we're talking about the cell membrane or the plasma membrane, it's just not phospholipids. Okay, they have other things that can be associated with it. All right, and the function of the plasma membrane is not limited to just being a boundary or physical separation. The plasma membrane has other very important functions, such as regulating what can go across it into and out of cells. All right, but like I said, there are more things associated with the plasma membrane than just phospholipids. All right, so several molecules can be embedded within or attached to the lipid bilayer, such things as cholesterol, proteins, glycoproteins, and glycolipids. All right, and because that is more than just phospholipids, the plasma membrane is said to be a fluid mosaic. All right, because those lipid bilayers can flow across one another, and a mosaic because you have a mixed composition of molecules. You have the phospholipids, you have the cholesterol, the proteins, glycoproteins, glycolipids. And so because of that, it's a mosaic. All right. So here's, you have your phospholipids. Now these lipid bilayers can move across one another. Okay. All right. And so that's why it's called, the fluid part. And then the mosaic part comes from, you have all different types of molecules that make up the plasma membrane. And there are different types of proteins associated with the cell membrane that have different functions. You have adhesion proteins, all right, that help cells stick together. You have recognition proteins, right, that will be on the outside of the cell, and it can help the body or other cells recognize cells that should be there, recognize the self. Okay, and so these recognition proteins can help your other cells to recognize a cell within your body is like, hey, this is, my cell, it should be here. You have receptor proteins that when they are associated with some molecule, it can trigger a change inside the cell. Okay, so if a molecule from the outside of the cell comes and binds to that receptor protein, okay, through a series of chemical reactions, things like that, it can trigger a change inside the cell. And then you have transport proteins, and these proteins assist the movement of ions and molecules across the membrane that wouldn't be able to otherwise. All right, so there are two big categories of cells. You have your prokaryotic cells and your eukaryotic cells. All right, so these are three big differences between the two groups. Prokaryotic cells are smaller. All right, so now you can see them with a light microscope, but you're going to have to use a more higher power objective lens than a eukaryotic cell. Our prokaryotic cells don't have true nuclei. Okay, like eukaryotic cells do. Right? And so that's what houses the DNA in eukaryotic cells, those true nuclei. Now, in prokaryotic cells, the DNA is housed within a region called the nucleoid region, but it's not a true nucleus. All right, and so that's what the whole name comes from. So these two letters U, EU, all right, that's a prefix that means true, and then karyotic or karyote basically refers to the nucleus. Okay, so it's a true nucleus, and then before nucleus. Okay, so no true nucleus, and then the prokaryotic cells, in addition to not having a true nucleus, they don't have membrane-bound organelles, whereas eukaryotic cells do. All right, so let's go over prokaryotic cells. So what are examples of prokaryotic cells? All bacteria and all archaea organisms are single-celled organisms, and they are considered prokaryotes. Now, they were once formerly grouped together in the prokaryote kingdom, but now they have been separated apart. Okay, and so archaea, in fact, are more closely related to eukaryotes than they are to bacteria, even though the archaea and bacteria are both prokaryotic organisms. Right, and so because they're so different, archaea now have their own separate domains. So you now have three. You have the bacteria, archaea, and then eukarya as the three major domains. All right, so they don't have true nuclei, but the DNA is concentrated in an area termed the nucleoid region. When you get outside the plasma membrane, remember they're cells, so they, like all other cells, they have a plasma membrane. But outside the plasma membrane and a cell wall, okay, is a capsule in which many prokaryotic cells have, okay, and that capsule has sticky sugars or polysaccharides help aid in adhesion and protection. Okay, so not all prokaryotic organisms have this capsule, but many do. All right, and like I said, it lays outside the cell or the plasma membrane in the cell wall. Now, prokaryotes are said to have ribosomes, and we'll talk about ribosomes in eukaryotic organisms. They're not the same type of ribosomes. Okay, the prokaryotic ribosomes are different than eukaryotic ribosomes, even though they still sort of function the same in helping to create proteins. Prokaryotic organisms are also said to have or could have plasmid DNA. Okay, and plasmid DNA is just small circular DNA that they can get from different sources. Okay, that is DNA. So here in this photo, here's plasmid DNA. Now this is in addition to their normal DNA, but the plasmid DNA can give them benefits such as developing antibiotic resistance. They have flagella, which is this whip-like structure that helps in locomotion. But again, it's different than the flagella that we'll see in eukaryotic organisms. And they can also have pili, which are these cellular extensions that can help aid in cellular communication. All right, so now we're going to focus on eukaryotic organisms, okay? All right, and so what we're going to do is just kind of go through and talk about the major organelles that you can find in a eukaryotic organisms. All right, some of these are just in plant cells. Some of these may be only in animal cells, and we'll talk about the difference between animals and plant cells at the end. So first we'll talk about the nucleus and nucleolus. Okay, so the main function of the nucleus is that it protects the DNA from metabolic processes of the cell. Excuse me. So it houses and protects the DNA. The nucleus has its own sort of membrane, has a nuclear envelope, which is a lipid bilayer, just like the plasma membrane. Okay, so the envelope consists of lipid bilayers and a number of pores. Okay, which allow the movement of RNA outside the nucleus and proteins inside the nucleus. Okay, so you see here in this purple, this nuclear envelope, you see the holes, which does allow some things to go to leave the nucleus and some things to go in to help it in its normal function. You have a region inside the nucleus where the DNA and proteins congregate, and that's the nucleolus, and that's the location of ribosome subunit synthesis. Okay, so the nucleolus region, all right, that you see right here, that's where the ribosomes are produced. All right, and then the nucleus has a dense fluid inside. It's called the nucleoplasm. All right, which is similar to the cytoplasm that we'll talk about of the cell, and the DNA is suspended within the nucleoplasm. All right, and that DNA sometimes is organized as chromosomes, but most of the time it's more just sort of uncondensed, okay, and exists as chromatin. Okay, so when the DNA becomes condensed, there is considered chromosomes, and when it's uncondensed, it's considered chromatin. All right, so we will stop right there, and I'll pick up this next part on the next part of the lecture.